Okay, let's do this. All right. Happy anniversary, WebEVTalk for the second year today. So EV enthusiasts, welcome, and thank you for joining us for this special session of WebEVTalk. So some of you probably already know that we started during the pandemic, but when is that exactly? So let's go back a little bit. So the first time that the idea of a regular virtual meeting was discussed with EV communities on the 18th March 2020, which is perfectly like 2 years from tomorrow. And on the 23rd March, there's a brainstorming with Jan on the concept and format, and where actually the first time that we think about WebEVTalk as a name for this particular series. And on the 26th March 2020, we have the first episode of WebEVTalk with Jan himself as the speaker. So now, where are we? We are on the 2022. We have over 4,400 social media followers, which is across the Twitter, the Facebook, YouTube, and LinkedIn. And we have over 100 episodes, which comprise of senior and young scientists. And we have 50% female and male speakers. And in YouTube, we have close to 36,700 YouTube views, over 1,000 subscribers, reaching global audience, and including 44 countries. The top 3 is the US, Australia, and Italy. So WebEVTalk covers multiple topics, including the technology, isolation, imaging, characterizations, and so on. But disease, the role of EV in disease, in cancer, infectious disease, neurodegenerative disease, and more. We have also sessions on biomarkers and therapeutic approach using EVs. We have molecular biology, including the biogenesis, the characterization of heterogeneity, and so on. And we have biodistribution, in vivo imaging, and also cross-species communication, which is quite interesting nowadays. So today, we have this special session. We have someone who does not need an introduction, actually, Jan Lötvall, who's editor-in-chief in JEF. He's a professor at the University of Gothenburg, Sweden, and he was the first president of the International Society of Extracellular Vesicles. And he has published many papers in the field of EVs, including Valadi et al. Paper in 2007 that first demonstrated the EV-mediated RNA cargo delivery in vivo. And also, it's my pleasure to have Simon Powis. He is the deputy editor of JEF, a reader in immunology at the University of St. Andrews in Scotland, where he has been based since 2005. Simon is a biochemical immunologist who graduated from the University of Glasgow and Cambridge University. His current work is focused on the HLA class 1 immunopeptidome of EVs in health and disease. So this special session will be an interactive session led by Jan and Simon. It's entitled Navigating Publishing in JEF, the Intricate Work of Editorial Structure. So also we have Cecilia here, who's part of the editorial board in JEF that will be joining us today. So I now hand it over to Jan to lead our discussion. I'm just going to stop this share.
Thank you so much, Carolina. And of course, Simon is stuck in traffic, but I'm sure we can function anyway. So the idea of this session is no slides are going to be presented. It's going to be interactive. We will let you guys ask questions throughout the session and comment on the way we work with the journal. But as you know, I'm the editor-in-chief since 1st of August 2019. But I also helped start the journal in 2012. So actually, JEV is having a 10-year anniversary in only 1 month. So that's quite interesting. And I should say that there was a discussion. We started Journal for Extracellular Vesicles, or the International Society for Extracellular Vesicles, in the summer or fall of 2011, after the Paris meeting had occurred in that Graça Raposo and Clotilde Thierry arranged. At that meeting, we decided we needed a society, and then we had an interim board that started in the fall of 11. We started planning for a scientific meeting, which ended up being in Gothenburg in Sweden, where I'm situated, and I and Cecilia, who's part of the panel today, were actively arranging and helping out with that meeting, which was quite interesting. I should say about the meeting that we had about 500 delegates, but the biggest meeting room we had access to was 360 seats only. So we got quite nervous when we approached 450 registrations, and then we found out we could send video between different meetings rooms. So we started to accept registrations all the way until we have reached 500, and then we actually stopped the registration. We just couldn't take anymore. And that was a surprisingly high number of delegates, of course. At the meeting in Paris, there were 200. It was also oversubscribed. 200 people in Paris in was it January or February in 11? And we had almost 500 people attending in Gothenburg. But in the process of sort of starting the community, starting the society, we discussed whether we should start a journal as well. And I can tell you already now that there were some people in the board at that time that were very skeptical about us starting a extracellular vesicle focused journal, because basically their view was that EV research should be published in general cell biology journals, and that was some of the use. But end of the day, there was a majority of board members that agreed that we should start a journal, and we did start a journal. We published our first papers in late April, at the same day as we had the meeting in Gothenburg, this first day of the meeting in Gothenburg, and I think we had 8 papers in the first issue. Some of those are very highly cited, I can say already now. So those were from our group and from Clotilde's group and Yongsong Go's group in Korea and several other groups. And we got help from a publisher, which was a small publisher called Co-Action, which was a Swedish publishing group that basically was started as a startup by some librarians, I would say, entrepreneurs, and they had a series of different journals in their portfolio, including then JEV. And we published quite a fair number of papers in the first few years. Some of the niche work in those days, that's 10 years ago, the niche work, like, which centrifugation steps you should be using for optimized isolation of extracellular vesicles, that was not so easy to publish in other more general journals. But JEV loved that, and the community loved that work, and some of the work. Rosella is here. We also had a paper with Rosella in the early times on RNA content in extracellular vesicle subpopulations, and looking at different centrifugation steps and see how the RNA profiles were different. And those papers were quite extensively, or still quite extensively cited. So clearly the community has been quite excited about the journal. I think a lot of us have felt that it's been a struggle to get the journal into quite easy to get it into PubMed. After we had 10 or 20 papers published, PubMed access at that time was not so complicated. but getting us an impact factor, which unfortunately is a requirement by many researchers, took a long time. So and in that journey, coaction was actually sold to Taylor and Francis. So we were a journal within the Taylor and Francis portfolio for a number of years, with some issues and some well-functioning components. And then after the end of the contract, we were looking at a new publishing platform, and then we're currently associated with Wiley. But I should say that ICEV owns the journal, and that was something that was very important from the very beginning. So after the first issue, which I helped coordinate together with some other people, I was never formally maybe editor-in-chief or anything like that, but I was maybe founding editor. We had three. The board decided, actually without any application process at the time, to have a triple editorial-in-chief group, which was Peter Quisenberry, Yongsong Go from South Korea, Peter Quisenberry is from the United States, and Clotilde Terry, who's actually ending up doing a lot of the coordinating work for a long time. And this team were editors-in-chief all the time for I think 7 years, until I took over, or longer than 7 years, until I took over in August 2019. So I think in the early days before we had the impact factor, a lot of the work that came from the community and was intended for the community, and was probably work that we as a community otherwise had issues publishing in general journals, and it was directed towards the community, the issues we have, the problems we have understanding, topology of vesicles, or RNA content, or in general out of extracellular vesicles. So that was really, I think, has been a real help to the community, and it has helped us improve the quality of our science over the last 10 years. And then we had an impact factor in 2019. I mean, Cecilia, you'll have to help me with the numbers now, but I think it was 11 to begin with. The first impact factor was 11. And in 2020, when I taken over, it was 14.9 or something like that. And currently it's approximately 25, which is not our intention. It's not any goal that we have. We actually don't really care about the impact factor at all. But this has fundamentally changed the way we are functioning as an editorial team, and also changed the profile of the work that is submitted to the journal. So a lot of other people, especially from big countries in Asia, very big countries in Asia, like high impact journals, we get a lot of submissions from China. Some of it is great, and some of it is not great, but that's a fact, and there are high numbers coming in from that country, but also from other countries. So we built a editorial structure, or I have built this editorial structure to help manage the journal, which initially was basically myself as editor-in-chief, and then we have now a deputy editor team, which is it's Mary Bebawi in Sydney. It's Hubert Yin from Beijing. It's Simon Powis, who now has come through the traffic and is present in the panel, and Michael Freeman in Los Angeles. So and what happens is that what have happened initially was that I filter a lot of the work and look at it and sent a lot of stuff through to the deputy editors, but I gave my own judgment saying, this is probably not helpful. This is probably helpful for the community. And then, so we try to have two pairs of eye look at almost everything that we submit. But after a while, and we realized that it actually, the volume of submissions is huge currently. How big is it, Cecilia? Like 700 a year, 800 a year?
Yeah, something like that.
So I discussed with the board, ISO board, that we should have somebody who's working at least 50% for the journal, managing the journal. Firstly, I'm not good at policing, for example. You need to police all the submissions that come in to the journal, and how the editorial structure, associate editors are dealing with it, whether they are on top of everything and get things, what should I say, processed in a timely fashion, because it's all about time. It's about quality of editorial work, and it's about time. Of course, we need to be as quick as possible. So Cecilia came on board in May last year, and has been working half-time for the journal since then, through a contract with my university. Most of you know that Cecilia was actually my PhD student, and she graduated in 2013, did a brief postdoc in Japan, and is now back in the lab and doing very, very well in the lab. So that's the situation right now. And the way we've been working is that Cecilia screens the submissions. So she's pair of eyes number 1 currently. And then I have meetings with her, or one of the deputy editors will have meetings with her to go through those and judge whether they're worthy of further processing. So we do take a lot of decisions at the editorial level. Now, I'd like to give the word over to Cecilia for a while and tell us a little bit about how you deal with these manuscripts.
Yeah, so we get maybe 15 manuscripts a week, and I read all of them, and of course look at several things. The topic, if it's suitable for JEV, also, but I'm also checking, I mean, what we think is important is that the isolation of the EVs is solid, because we are an EV journal, and we should publish things that we are certain that the function that is reported, or the marker that is reported, are truly EV associated. So go through the method sections to make sure that the EVs have been isolated in a good way, that they are characterized, that we actually know that EVs have been isolated. So I go through all these different parts of the manuscript, and then twice a week I have a meeting with Jan or some of the deputy editors where we discuss it. So we are always two people who make the call on whether this is a manuscript that we should process in the journal. Then we send it forward to some of the associate editors to invite reviewers and so on, or if these manuscripts should be rejected straight away, either based on that the topic might not be suitable right now for the journal, or if the EV part is just a small part of the manuscript, or if the isolation and characterization is not up to standard, or we don't think that the claims can be supported that is truly an EV-mediated function or marker that is described. So we're always two people discussing every manuscript, but I'm reading them all to make sure that, because we also want to save time. So it's better to get a quick rejection than going through a longer processes and then might be rejected in the end anyway. But also with 700 to 800 manuscripts a year, we don't want to exhaust the reviewers on things that we might think that will not make it in the end anyway. So we try to be quite selective in this early phase as well, to let things through that we actually think is important for the community and have a good standard. So that's basically the process that we deal with all incoming manuscripts.
Yeah, and then either I assign a deputy editor, or when you have the meetings with the deputy editor will decide the deputy editor. So Simon, how do you find this workflow, and explain a little bit how you work with this? I can say that you're the fastest deputy editor. You know, you're faster than your own shadow. And it's in dealing with manuscripts. So, but tell us a little bit how you deal with it.
Yeah, my apologies for being late. They seem to be digging up every single road entrance into St Andrews at the moment, so it's just impossible to get in the morning. So my apologies for that. In terms of being fast, it's also due to short memory. If I don't do it straight away, it just flies out my mind and I forget to do it. But so the system that Jana was referring to and Cecilia was referring to basically means that one of the 4 deputy editors has recently been doing periods of a couple of weeks and going over the manuscripts. Like Cecilia, when we get the manuscripts, I will read the manuscripts as fast as I can, go over them. And again, we focus on some of the things that have been mentioned in terms of methodologies and overall potential impact of the results. And then we will pass those on to associate editors. I think there's about 20 to 25 at the moment. And then we will, there's a standard format of letter that goes out to the associate editor saying, please invite reviewers. We will maybe adapt that letter and add in a few comments to say, you know, we think this is important because X, Y, and Z. And perhaps we might consider such and such a person as a good reviewer if we've got someone in mind that we might think appropriately fits the submission of the work. And then we'll encourage and try and keep things ticking along as fast as we possibly can. I think because of the pandemic especially, and the time pressures everyone is under, reviewers are often a bit slower than we'd love to have, but I think that's the case for all journals that are occurring at the moment. So when you submit something, just be prepared that, you know, the review process might take a little bit longer than would be absolutely ideal, but I think that's a circumstance for many, many journals. Thanks for that. Yeah, happy to add comments or if you've got any specific questions.
I would say that if your manuscripts go through and it reaches an associate editor, the likelihood of it being of interest for the journal is much higher, right? It's clearly, I think, roughly we discard or decline maybe more than 50% to begin with. So maybe there, I mean, I'm taking, say, rough numbers now, but maybe we reject 60%, Cecilia, or
yeah, probably
70?
maybe even more. We reject a lot in the first step. Yeah.
So, and then probably 50% go through after the editorial process, but there are some rejections, of course, especially if there's lack of novelty and so on. So, and when we look at priority of publications, my attitude is that JEV is a journal for the EV community. So if there is a paper or submission on review, for example, of extracellular vesicles in rheumatoid arthritis, that's not for us, right? It could be the most brilliant review article that you imagine, but it should really reach the rheumatology arthritis community. And you know, we might get a lot of citations with a review like that, but I feel that we should let those important publications reach the right audience. I don't think that rheumatology scientists are necessarily primarily looking at JEV, but actually on their own journals and following their own journals in detail. And I think that's probably a better general strategy. So that's one way we are thinking. Another, so that was an example of a review article. If we look at original papers, we can get studies on the role of microRNA X, Y, and Z in acute liver failure and the role of exosomes or something like that, right? And most of those are probably declined because the focus of the paper is the disease. And the focus of the paper is not the EVs, right? And we are not a acute liver failure journal. We are an EV journal. So there has to be some additional understanding of how the EV and what in the EV does something for JEV to get excited about this. So that's something that we consider in this process. And there's of course a gray zone between, pure disease model data and nerdy EV molecular biology research, right? And there's a gray zone, and we're not super exact maybe in our judgments there, but I think that's something when you submit to JEV, that's something you should think about. So the title of the paper is exceptionally important. If it's all about a disease and the excitement communicated in the title and in the abstracts is around the disease, it's probably intended for another type of journal. If the excitement or the focus and the interest is in around the nitty-gritty of the EVs, yeah, we love it. And then it's most likely or more likely to be considered by the journal. So also I should say that, and you mentioned that, Cecilia, we do not consider at all studies that purely isolate vesicles by kits. So that's we know that no kit is perfect. So the isolation can be supported by kits, and we acknowledge that and we understand that. But pure kit like precipitation kits, for example, we know they carry bring a lot of contaminants into the isolates, and that's not necessarily what we want to study. Now, we should also mention that there's a new journal that just published its first papers, Journal of Extracellular Biology from ICEV, and I would call it a sister journal, not a journal that we put other or things that are not accepted by JEV and automatically goes to JEXBio. That's not the case at all, and JEXBio will have its own evolution and so on. It's not totally focused on EVs, and I think maybe next time we should have a meeting with Andy Hill, who's the editor-in-chief for that journal. But we do send some papers to, or suggest the authors rather, to consider submitting their work to JEXBio. And once the manuscript is in the submission system, it's quite easy to do that. So I don't know how many. Do you know how many papers how many we have suggested to be transferred to JEXBio and where the authors have considered to accept that, Cecilia?
No, not on top of my head, but basically what's happening is that JEV would reject the manuscript and then suggest that you could transfer it to JEXBio, but that would be up to the author to actually do that. So I know that, of course, some people, submitted something to a journal with 25 in impact factor and will then not accept a journal with no impact factor yet. So but some people have accepted it, and I think are in the process, in the system now for GexBio. And so, but I don't have an exact number now.
But we should acknowledge here that that's where Jev started 10 years
10 years ago, we didn't have an impact factor, and nobody wanted to, well, no, some people didn't want to publish in a journal without an impact factor then as well.
And of course, you, the community, made the journal because you submitted the cool work to Jev. So that was really, really helpful.
I think it's super important to support the journals in the beginning as well. That's the foundation for the first impact factor, which was 11 or something. So it really indicated that those papers were highly appreciated by the field.
So somebody asked something in the chat here for Maimona. You can ask a question verbally. If you straight up your hand, and we will unmute you. Hold up your hand. Where I don't see you because I don't have everybody on.
Hi, yeah, Maimona here.
Yeah,
thank you so much for the effort.
Yeah.
So I'm not able to open the video now, but thank you so much for this chance to discuss about Jev and EVs. And as newcomer to the field, we really appreciate your efforts to raise the standards and the quality of the published studies in Jev. So for us, Jev is a reference, because like when a newcomer come to the field, the quality of the published studies everywhere is not up to the standards. So anything could be claimed to be EVs, anything could be claimed to be microvesicles. But Jev, stand out, and it's a reference for us, new researchers. So I was asking about the reviews that assess the quality of the studies, of the published studies, and the reviews on use of EVs as therapy for specific kind of diseases. Because as you mentioned, this is not for Jev, right? Or for the journal. But then if you approach other journals,
it can be. But go ahead.
We recently sent a review. It was a very extensive and comprehensive on how some studies, it's like a specific field, the use of EVs in treating some conditions. And our focus was on the quality of the studies, whether they followed MySafe, they followed the guidelines, and their quality of reporting as well. So it was very extensive on this issue. And we think if we submit it to another journal, maybe they might not be that interested because they are not EV people. But then we faced this, the journal was not interested in this review. The reason is because it's the focus was the disease, the condition. And so that's why I'm asking here, because I think you just mentioned this. So if this kind of studies that focus on the quality and rigor in research, because as I mentioned, as a newcomer to the field, when I first started to read the papers and publications, I faced this problem of not knowing how to do the good quality research. And then if you start with the research that's not of good quality, then at the end, you're not getting what you're looking for. So so whose role to focus on this, or to publish this kind of studies?
So I'll just give you immediate feedback. It's all about how you write it. If you write it around the disease, then of course it's about the disease. Now, if you write it about rigor of EV experimental therapeutics research, and discuss your findings in your I guess structured search and so on, and that's a different matter. But it's really, if you start your sentence about there's a big unmet need in understanding the effect of EVs in rheumatoid arthritis. I'll keep staying with that. Then your article is about rheumatoid arthritis. Now, if you start your sentence or your abstract saying that there's a big unmet need in understanding the rigor of therapeutic in experimental therapeutic studies using EVs, especially, and then you start discussing those issues, that's a different paper, and it's really down to the sentences that you write and the title, right? So think about that when you write your papers and submit it to Jeff. That's what I'm trying to say. Does it make sense? Do you
Yeah,
agree,
so now
Simon?
I guess, yeah, maybe
go
the
ahead.
way we phrased the title and everything was the focus on the disease, but yeah, it was a systematic review. So we assessed the quality before drawing the conclusions from the studies. So yeah, now I get your point. Yeah, okay. Yeah, thanks a lot.
You agree, Simon?
Yeah, absolutely, yes. And the latter point is about is raised there about the adherence to MySEF guidelines, which is a very relevant point as well. And I think it's worth us saying that, we do not strictly adhere absolutely to MySEF guidelines. We want people to do the best characterization they can, but we recognize that not everyone can do anything because not every laboratory has access to every single type of, characterization technology. Like in our case, we had a building fire, which just taken out our electron microscopy. So we can't do any electron microscopy at the moment. So yeah, we recognize that not every lab can do every single technique, but nevertheless do the best you can, and we will consider what to do.
Yeah, and also there may be techniques that are not discussed in the MySEF guidelines and new techniques that we have to consider that might be even better or than old techniques. But what we do expect from
just
people,
to keep the minimum, just to check
Yeah.
if they, for example, check the size. If they claim it's small extracellular vesicles, they need at least to check the size of their preparation, and also the positive controls and negative controls. So for us, we divided into two parts. One part is for the finding of studies that not really comply to MySEF. And then we had some study on a small part on the studies that complied to the characterization of MySEF. And then we had some interesting findings on that. So but maybe the way we phrased it was basically mostly focusing on the disease. But it's good to know. It's good to know, actually, maybe for our future reviews. So but we understand not everybody has the facilities to do everything, but just to keep at least the minimum, you identify your preparation, whether it's pure enough, the level of purity, and whether it has some of the markers that are advised by MySEF. I think MySEF is already minimal, it's the minimal requirements to identify the preparations, right?
But it's guidance, and as Simon said, we're not absolutely requiring everything, and it can be different slightly to what MySEF does. But what I do think is important is that the MySEF is considered. MySEF is considered, and also perhaps even more importantly, EV-Track. Which describes the exact antibodies and the diligence of the reporting of the methods is also important to consider. Kendra, you had a comment or question?
Yeah, more or less along the line. It's really great from you to hear the scope and that you're mostly directing the EV community. Is there a chance, when we're now preparing a manuscript, to talk to somebody before setting the focus, if it's a general interest of Jeff, or if it's rather should go to a more focused audience?
Yeah, you think about it when you write your manuscript, exactly. If you look at, go ahead.
Yeah, the question would rather be, so now we are in the process of preparing the manuscript to get a feedback beforehand, if the general topic would be of interest.
Mm-hmm.
Is there a chance to talk
and
to
you can send pre-submission inquiries to eic.jev@icev.org. So maybe we can put it in the chat, but it's editor-in-chief, eic.jev@icev.org. Should be easy to remember once I've said it, maybe. Any more comments or questions?
I just want to follow up.
Has asked a question.
Oh, sorry. I just want to follow up on what Jan said before with the EV track and the method sections, that if you submit something to JEV and you just write, we isolated vesicles according to this reference, full stop, that is not sufficient. You have to describe, because you never do exactly as something else, and then you would, the reader or the reviewer would have to find that paper, and then sometimes in that paper the method section is in the supplementary method, and it takes us quite a lot of time to understand how you isolated your vesicles. And we really support that when somebody read a paper in JEV, that they can easily evaluate if they support the claims that you're making. So if you have like a molecule that you're saying doing something in the EV, you should probably have done a density gradient, for example, to make sure that this protein is actually associated with the EVs and not just soluble protein pelleted during the ultracentrifugation and so on. So really consider the method sections when you write your manuscripts as well. And then I think Simon wanted to highlight that Jenny asked a question.
Yeah, Cecilia, have you got any idea at the moment as how much time it takes to get first reviews back? My feeling is about a month averaging at the moment.
Yeah, I mean, you have to understand that, like, first the manuscript is submitted. It actually goes to a quality control check. So that's done by Wiley. So it will take a few days before it reaches Jan and me and the deputy editors. And then, of course, we meet twice a week. So it takes a few days before the reviewers can be invited. It's not like you submit your manuscript and then boom, 2 hours later, we invited reviewers. It has to go through the quality control with and so on. And then, of course, it goes to the associate editor. They invite reviewers. Normally, we give reviewers 2 weeks, but they're not always accepting day one. They can wait a week with accepting, and then they all of a sudden the 2 weeks starts from that. And of course, sometimes more time is needed. Sometimes they decline. We invite new ones. So you shouldn't think that you can get a decision after 2 weeks just because that's what normally reviewers get in time. So of course we're trying to be as fast as possible, but we have also changed the system. Maybe you've seen if you submitted, we changed from editorial manager to Scholar One, and there have been some delays due to that process in the last 6 months. But so yeah, it 1.5 months, I would say, before you can get the reviewer comments, probably.
There is I missed that question in the chat, but I agree. It takes probably a week before the associate editor starts inviting, and that's not bad. So I don't have the absolutely latest statistics, but usually time to first decision on average is 30 days, and for general journals in general, and we are well within that in our statistics. So, but of course that includes a lot of decisions where we early decline, and that's part of the statistics. So the median and some papers can take a long time, unfortunately.
And
Carolina,
I mean, if sorry, I just want to, if you're invited as a reviewer, please accept, because obviously we thought you were expert on the topic and we want your opinion. But if you decline, I mean, please do so quite fast then. Don't wait for weeks to decline, because the faster you decline, the faster we can invite somebody else instead. So this is also a process that we are depending on, and we understand that everybody's doing this on their free time, and we're really grateful, but this is a process that we can't always control.
And before I get to Carolina, I should emphasize that when you submit your work, you get 2 reviews back. 2 people have really gotten into and trying to help you increase the quality of your work. Please return that favor to other authors. So for every paper you submit, you should at least review 2 more papers, right? Or manuscripts. So maybe even more. So Carolina, please go ahead.
Yeah, I just want to add into that questions from Kendra. I think one of the way to actually get some attention from Jeff editors is to actually have it published at the preprint, and you can also present at WebEVTalk, and then you likely get some more attention from Jeff editors. But I also have a question here. So with the massive submission that you received so far, can you sense on the trend in terms of topics? Where does it goes? What is the most interesting topics going to be for Jeff?
Simon, you want to comment on that?
It's a really tricky one. The quality of the papers is rising for sure, and the ones that are coming through to me as handle as deputy editor, I'm very impressed. The ones I've had this week, I just thought absolutely astonishing. Some really interesting data. I can't sense an overall trend at the moment, though. Perhaps Jan and Cecilia might sense a trend. I can't.
Cecilia?
I mean, I guess it's a different trend. One trend is what people submit, I guess, and one trend is what we're interested in and letting through.
Yeah, I think more what you letting through, I think, so we know as a person who submitted to Jeff.
Yeah, so to follow up on what Jan said before, sometimes we can get comments maybe that, oh, this paper is just comparing different isolation methods. This is not for an impact factor 25 journal. And we disagree there. We think that what the EV community is interested in is comparing isolation methods or comparing characterization methods, evaluating FBS contaminations, or all these kind of basic things are actually things that we think that fits quite well in JEV, and is most likely not a topic that other journals would be interested in because they are more disease specific, for example. So that kind of papers we are highly interested in, but it has to be done in a good way. Like if you want to claim that something is better than something else, you would really have to dig into that and look at that in a good way. And when it comes to, of course, we are interested in papers regarding some diseases as well, but then you kind of have to have the understanding that you really have to prove that the molecule, whether it's a microRNA or a protein, are inside the vesicle or a part of the vesicle. You have to prove that. You also have to prove that which molecule in the EV are doing the thing that you see in the recipient cell. Just taking EV, giving it to a recipient cell, and have half a paper on what's happening in the recipient cell and the disease is not necessarily our focus, because we want to know what in the EV did that. So as Jan said before, it's the EV focus.
Yeah. So how do you see protein corona of EVs then? It's not exactly inside the vesicles. Can we still publish those sort of papers?
Yeah, I think we have maybe some papers on corona. I mean, it's not something that we would reject straight away. We think that that's interesting to understand the coronas of the EVs, for sure. But I'm just saying that I also see a question here on isolation kits. And for example, some people might just use an isolation kit, no characterization of the EVs, claim some molecules are doing something in the recipient cell. Of course, that's not enough for us, but if you can prove that this molecule, whether it's on the inside, outside membrane, is transported by the vesicle, we're interested in that, but it would have to be the EV focus.
Thank you.
What other methods beyond kits? I mean, we do accept papers with ultracentrifugation, kind of sequential ultracentrifugation approaches. But the gold, if you're working with in vitro, the golden standard is often to use a density gradient somehow, or an OptiPrep cushion or something like that. And that's a golden standard. There are other methods, and we do accept other methods, tangential flow filtration with concentration steps, for example, as long as your EV isolates are well characterized. So, but just kit, just a PEG-based kit, it's not going to cut it. That's not really. There's so much contamination, and far more contamination than the corona of the vesicles. Anna.
Hi, thanks a lot. So I'm just curious if you could talk a bit more about the difference or where you would like to see the difference between JEV and JEXBio. I think maybe this is also related to these questions.
I think that question should be asked Andy Hill. Of course, ICEV is still seeing JEV as the sort of crown jewel, if you may, of ICEV. It's a very high impact journal currently. By the way, we do expect the impact factor to go down significantly, and I'm not worried about it at all. I hope you're not either. But anyway, I think Andy will be more open to work that is not exceptionally detailed or to the exceptional standards that we might request or expect in JEV, but also, if I understand correctly, that journal will also look beyond extracellular vesicles. I mean, we're not necessarily not going to do that either, but beyond what's exomers and other things that may be in the interstitial space. So how JEXBio evolves, you have to remember one thing, a really important one. A journal is only the quality of its submissions. So you decide the quality of the journal in a way. Remember that. And editors can only manage what we get in. We don't create the quality of the journal. We create the quality of the assessment of manuscripts, but we don't create the quality of the publications. You do that. So same thing with JEXBio. We'll see how it evolves, I think. Nada?
Yeah, hi, Jan. Thank you very much. Just following up on my question in the chat about the isolation and everything. So I think my question is mainly about how would you accept it when it's mainly from patients, like patient blood, for example. So a sample that's inherently, impure, and especially when you're looking on larger EVs, so not necessarily doing ultracentrifugation and stuff like that. Just wanted to ask, because I haven't seen much publications recently about larger EVs in JEV, so I just wanted to ask how.
Cecilia, you want to talk about patient samples? We have a manuscript that is now accepted, Cecilia, or I'm not sure.
No, not yet.
Okay, we shouldn't. Okay, so we've been working on purifying and isolating vesicles from plasma and serum and comparing the two types of vesicles. It is HELL. It's such hard work, and Cecilia Nasib in our lab has been working on that for a long time, and it was much harder. the original sampling is very important. The contamination of platelets is much bigger than you think. So yeah, blood samples from humans is really hard, and using a kit that says it isolates vesicles from plasma from patients is just wrong, right? It doesn't mean that the data is uninteresting. The isolate may contain something that is important in RNA or what have you, but we cannot say that that is a vesicle. That would be not, would not, the conclusions would not be supported by the data. So it's
yeah, I mean, a biomarker is always a biomarker. It's interesting,
exactly.
But is it an EV associated biomarker is what we ask ourselves when we get the manuscripts to Jev. And to follow up on your question, we are equally interested, I guess, in large EVs as small EVs. It's not like we are favoring exosomes or small EVs over microvesicle and large EVs, but we get much more manuscripts submitted.
It's the amount of amount of studies.
It's less on larger EVs, I would say. Yeah,
I guess that's because people use UC, ultracentrifugation, and 100,000 G, and then call them. There's some opinion that those should be called small extracellular vesicles. I'm a bit concerned about that nomenclature because you have big vesicles there as well. And the overlap between big and small vesicles is enormous. It's really hugely overlapping. On average, you have smaller or bigger, depending on how you do your isolation, but it's not like there's a distinction between those. There's no clear-cut distinction between those.
Yeah, and I mean, that's another thing that, I mean, things that could be interesting for Jev is that if you're only looking at one type of vesicles and claiming they are doing something, of course, it might be interesting if you also have other subpopulations of vesicles not doing that, for example. That kind of comparisons would be interesting as well, whether that's large and small, exosomes, microvesicles, or whatever cutoff you have, high density, low density, or so. Saying that one of them doing something but not the other one is actually also interesting, but a lot of papers are just focusing usually on one, and then we don't know how unique that is.
Another issue. Yeah, go ahead.
No, sorry to interrupt. Oh, thank you. It's just that, again, as Cecilia mentioned, like, this is a very interesting point, like different populations. But again, how pure should these populations be? And again, as you mentioned, how can we be sure that probably it contains other contaminants, especially if it's from human, like patient samples, blood specifically. So it's going to be, again, a mixture of other things other than vesicles. So it's a bit tricky to...
Then it's about how you conclude, right? If you conclude it's vesicles, then your data is not supported by, or the conclusion is not supported by the data. And that's the key. That's where the key is, right? So, your data might be fantastically interesting. And another thing I wanted to mention, that an issue that we as a community need to work better with. What is the control for an EV? What is a control for an EV? Is it a lysed EV, or is it a sonicated EV, or is it PBS, or what have you, right? So, Simon, you have any comments on that?
No, total mystery.
It's hard, right? I mean, I remember discussing this 10 years ago, almost, right. and it's still an issue that we have to deal with. I think Manuel has a question.
Hi, thank you. Yes. Well, I haven't ever submitted to GIF because of as well of the open access fees, but this is another history for another day. But related to the last comment of Cecilia, when I'm particularly focused on studying different population of EVs. So what would you need to prove that they have a different function or different activities in cells? So because you said, okay, we are comparing the population, demonstrating that we have different content, slightly different sizes, but then what you need to prove that they have a different bioactivity?
Yeah, so providing? I mean, that would be...
Let him answer it. Let him answer. What is the data you have?
What is that?
What is
Well,
the approach you're taking? We're not going to tell you how to do your research. That would be inappropriate.
I know.
Actually. Yeah.
I understand. The thing is that you said, okay, the focus is not on the effect, but it should be on the vesicles, right? So then it comes a little bit in this balance, not between, okay, I want to know, or I want to show that they have a different effect, but I do not want to focus on a particular cell or whatever. But then at the end, if it's not accepted or if it's rejected by Jeff, you need to go to another journal and you need to focus. So that's the balance that you need to always consider, no? If to focus on a particular field, let's say I'm working for regenerative tissue engineering and regenerative medicine, or focus more on the interest, on the differences. But then this problem comes, not that if it's rejected in Jeff, perhaps it's not interesting in other journals.
Yeah, so we are interested in subpopulations. Exclamation point. Yeah, we're definitely interested in subpopulations.
Okay.
And if you can show a different function of the different subpopulations, that's fast track.
Okay, I've been struggling to publish that.
Did you send it to Jeff?
No, because of the open access fees. As an emerging researcher, I didn't have access at that moment to pay for it. So that was not an option. Well, I tried to get the waiver, but was not enough.
Yeah, I know. Now this is how to train the universities of the world that we have to use open access, and we have to accept that it costs that much to publish a paper. It's not that it is, even if you look at papers that are published closed access, the on average the cost for a paper is $2,500, or was 10 years ago $2,500 in closed access journals, right? If you go to Nature, the diligence of the editorial review process is much more extensive. It's probably even more to publish in Nature or New England Journal of Medicine and things like that. But they're taking that money from the universities through other ways, right? Not through your specific grant, but through your overhead, which goes to the library.
Yeah, no, I completely agree. As I said, it's a history for another day.
Yeah, it's a topic for another day. It's true.
Thank you very much.
You had another question, Nada?
I'm sorry,
Comment?
just a quick one, because again, I'm interested in subpopulations, but I would say how pure, what size, how pure would you accept a subpopulation if it's from patient samples? Because I've seen publications, not in JEV, but as you mentioned, how would they really be sure that these are EVs that are doing these things? So how pure would you accept that, please? Thank you.
I mean, it's hard to draw a hard line in the sand and say this purity and not that. But in generally, I would say that a lot of people, when they characterize the vesicles, they pick 2 or 3 EV markers and to say they have EVs. But I think you should also consider non-EV markers. So to understand your contamination, you need to look for it. That's what I mean. So we did that for our first plasma paper a few years ago. We looked at the EV markers and we were like, great, our EVs are in this sec fraction. It looks great. And then we did EM and it looked terrible. We did proteomics. It was horrible. And then we also looked for contamination, so like the ApoE, because this was plasma. And we had a lot of ApoE in the EV fraction. But if we just look for the EV markers, we would say that this is the fraction we want to choose. So I think it's also important to understand your contamination. And then it's important to understand what is the contamination for the samples I'm having. So the markers for contamination is not the same for CSF as for plasma, as for breast milk, as for urine, as for cell culture. You have to adapt that to your particular starting material. That's the only way to understand that, okay, I have EVs, but what other things do I have? And I think that's really important to evaluate as well, because you can have EVs and you will get the positive marker, but what else do you have? You have to be curious about that as well.
We had a question earlier, what's the trend in submissions? And I would say that one trend that we see quite a lot is that people are claiming the role of some microRNAs in some disease model through a certain axis, and the word axis is in the title, and then one or two microRNAs are in the title, and the disease is in the title. Those are usually not really well, some of them could be okay and good, right? But some of them are not that diligent, and the data is not necessarily supported by, the conclusions are not necessarily as strongly supported by the data. So that's a trend we see, but it's a concern we have as well, that people are thinking a little bit too much on microRNAs in vesicles. There are many other things, and proteins are probably really important. We don't see so much about the lipidome either, so I think that would be interesting for us to get more publications in. So that's something to consider. I think we're coming towards an end, Carolina. I have something to say before we finish, right? So Cecilia has functioned now as half-time executive editor for JEV from 15th of May last year, and she will finish on the end of July this year. And from the 1st of August, there will be a new executive editor. And that job has been advertised by ICEV. And if some of you attending this meeting yourself, or you know somebody with some editorial experience and understanding of science, of course, and also of EV science, please consider applying, because it will give you a lot of insight into EV research. You'll see everything that comes through to JEV, but also to JexBio, because that role will include involvement in the JexBio editorial workflow. So I haven't seen the advertisement. Have you seen it, Cecilia? Is it on the Facebook?
People have shared it. So I think
Good.
ICEV have emailed about it as well, that this position is out.
Open.
Yeah.
So and it's not necessarily that you sit in the ICEV office, right? It's you can probably be located anywhere. So
I mean, it just happened that you and I are both in Gothenburg, but with the pandemic, we're having the meeting
we have
through
not had
Zoom
a single,
anyway.
we had not have a face-to-face meeting on this topic since we started this work. We've met face-to-face, but not for this. So okay. Any other last words? Otherwise, I think thank you very much, Carolina, for inviting us. She actually asked me to give a talk, and I said, why don't we have just an interactive meeting? I love these interactive meetings, and people can speak and talk, and we can have an open discussion and a small panel. So thank you everyone for participating, and I hope to see you soon again. And maybe next time we can, in a few months or half a year, we could have a meeting with Andy and myself as well, and we can talk about how JexBio and JEV has evolved. Thank you, Carolina.
Yeah, thank you so much, Jan, Cecilia, and Simon for the panel today and question and answer as well. It's a great time to meet you virtually. And Cecilia, good luck for the next 4 days. Just a short message that...
okay, yeah,
sorry.
Anna's not raising her hand. She's just clapping her hands or something. Yeah, sorry. I thought it was raising her hand. Yeah.
All right. Yeah, just a short message that if you end this meeting, there will be a survey coming up, which is about your ideas or suggestion for WebEVTalk. So please fill in that survey so we can do better next time. Okay. So until the next meeting, I hope everyone take care, and yeah. Hope to see you soon.
Bye, everyone.
Bye-bye. Thank you.
Thanks.
Thank you.